All right, hello everybody. My name is Lauren Lees and I'm a PhD student at the University of California, Irvine. And today I'm going to be talking to you about kelp microbe interactions and their role in mitigating nitrogen pollution. So today I'm going to be focusing on two kelps of interest that I study. Giant kelp, which you're probably familiar with, macrocystis. It's frequently found in Southern California, and it's usually what you think of when you think of kelp. And then I'm also going to be focusing on another species of kelp, the southern sea palm, or Eclinia arborea, which is a little bit different, but it's still a canopy-forming kelp found in Southern California. So these two kelps actually coexist. So they exist in the same location, but occupy separate niches. And what I mean by that is they're found in the same geographic location. So here we have the view back to Catalina from Bird Rock. And if we look closely, Eclonia will be found down here, right where at low tide, the water just touches the land. While macrocystis is out in deeper depths. So they're in the same area, but they don't compete with each other. Despite that, they both provide vital ecosystem services. So they sequester carbon through photosynthesis, removing CO2 from the air. They also form habitats for other organisms like fish and invertebrates. But particularly what I'm interested in is their ability to uptake nutrients, specifically nitrogen. Why do we care about nitrogen in the ocean though? So excess nitrogen is known to have harmful impacts, specifically harmful algal blooms. And if you were around Southern California in spring of 2020, you might remember this bloom that caused this really pretty bioluminescence. However, this was actually a harmful algal bloom that led to massive fish deaths and even caused human impacts. So surfers were coming out of this all itchy and feeling bad due to this harmful algae. And this nitrogen, this excessive nitrogen, can be found from naturally nutrient-dense waters that are brought up to the surface from a process called upwelling, but it can also be due to anthropogenic nitrogen pollution. And this can be a big problem in Southern California. We have over 20 million people living here, and those people produce wastewater effluent and contribute to terrestrial runoff that contributes to more nitrogen in our coastal waters. But it's not just total nitrogen that I'm interested in. There are different types. So first we have nitrate, and it's really found in that natural process again from upwelling, which brings really nutrient-dense waters up to the surface. But we also have ammonia, which can be naturally produced from animals, but it's also a big component of wastewater effluent. And lastly, we have urea, and that is a major component of terrestrial runoff. And so as you can see here, ammonium and urea are two major forms that are indicative of a large amount of nitrogen pollution in urban areas. And specifically, urea is interesting because it's an organic compound. And so if you'll notice this molecule up here, it's way more stable than the other two. So it's kind of harder to be broken down by organisms that live in the intertidal and the coastal ecosystems. But one organism that loves nitrogen is kelp. And so first I'm going to talk about giant kelp, and it loves nitrate. It's a fast-growing algae. There's a reason it's called giant kelp. It needs a lot of nitrogen to grow and photosynthesize. But if it can't get nitrate, it's happy to take up ammonium as well. It can take it up at pretty fast rates, not as much as nitrate, but it's still fun. And finally, it's actually been shown recently that it can take up urea at rates similar to that of ammonium when exposed to low nutrient conditions. The southern sea palm, on the other hand, is a little bit more mysterious and less is known about its specific nutrient dynamics. We know it prefers to take up nitrate like most macroalgae does, but it can also take up ammonium at high rates. Its capacity to take up urea is really understudied, and we don't know that much about it yet. But the kelps aren't doing this all on their own. Kelp is home to a really complex microbiome that lives on its surface. And these microbes can prevent colonization of kelp by pathogens. They can produce vitamins and micronutrients for algae to use. But what I'm interested in is the ability of these microbes to help the kelp take up these different forms of nitrogen that might be a little harder to usually take up. And microbes might be able to do this because they can change much more quickly than kelp can actually adapt to these changing conditions. And specifically, these complex forms of nitrogen might be able to be broken down by microbes much more easily since they have really diverse metabolic pathways. This leads me to the two main questions that I'm studying with my research at Catalina. Do the nitrogen dynamics where the kelps exist affect their ability to take up these different forms of nitrogen? So if we have one kelp that lives in an area that is really exposed to nitrogen pollution, how is that going to affect how fast it can take up these different forms of nitrogen, as opposed to a kelp that lives in a relatively unaffected area by nitrogen pollution, but is exposed to coastal upwelling? But like I said, it's not in a vacuum. So how does the kelp microbiome impact that nitrogen uptake? And does the actual microbiome change with those different local nitrogen dynamics? To test this, I'm using a geographic gradient that's exposed to different levels of nitrogen pollution. I have three regions that I'm currently testing. We have the front side of Catalina, where I look at Bird Rock and Big Fisherman's Cove. And these two sites are relatively removed from the large amount of urban nitrogen pollution that we see in the Los Angeles or Orange County coastline. But there's still a little bit of nitrogen pollution present. And then we have the backside of Catalina, which is the closest thing we're going to get to a clean site. It's relatively removed from the nitrogen pollution of the mainland, and it has a lot of naturally nutrient-dense waters from upwelling. And finally, I have two sites on the mainland that are exposed to different levels of nitrogen pollution. And this is the highest level of nitrogen pollution in my study. So at each of these sites, I collect kelps. And here we are in Big Fisherman's Cove collecting macrocystis. And then I take tissue samples for microbial analysis along the blade. And I use these tissue samples to get DNA and sequence a specific region of that DNA to know what microbes are actually there. Then I attempt to manually remove the microbiome from half of the blades using a sterile microscope slide. Finally, after the kelps have been allowed to kind of rest and recuperate after these different treatments have been applied to them, I measure the amount of the different forms of nitrogen that it can be taken up by these blades of kelp. So basically, I put one blade into each of these chambers here, and I spike in the target form of nitrogen in various levels. And then I take water samples every 15 minutes for 2 hours to see how much nitrogen is being taken up over a time period. You might be wondering, cool, what are those results? Well, this is an ongoing project, so unfortunately I don't have specific results to share with you today. But from my time at Catalina, I was able to take over 1,700 water samples that I froze and had to bring back to my home lab at UCI to analyze for those different forms of nitrogen. And along with that, I have 400 DNA samples that need to be extracted, sequenced, and analyzed. But I'm really excited that this project is finally off the ground after COVID, and I'm super pumped to see all of the preliminary data coming out of these analyses, and I'll be excited to share those with you too. But for now, that's all I have. So I want to say thank you. And if you have any questions, please feel free to email me. My email is right here, and I'd be happy to answer anything.